Hello and welcome to Lecture 3. In this lecture we are going to look at the process of gastrulation and how we move from bilaminar disc into the trilaminar disc. During this process we are going to uncover how our body is mapped to form a cranial and caudal axis, a left and right axis, and determining the locale of the organs. Let's have a recap of the processes we have seen so far and where we left the embryo at implantation. By the end of week 2, we have formed a bilaminar disc and we are fully implanted into the endometrium of the uterus. We consist of an epiblast layer and a hypoblast layer, which has helped to form a dorsal ventral axis. We shuffle dorsally into the wall of the uterus through the use of trophoblasts, which have begun to differentiate into cyto and syncytio trophoblasts. The latter of these are degrading the maternal capillaries and forming lacuna, which is the start of the utero-placental circulation. Extraembryonic mesoderm has developed and enveloped the chorionic cavity, and a small stalk of the extraembryonic mesoderm anchors the bilaminar disc to one edge of the cavity, allowing it to suspend. The syncytio trophoblasts are acting on the maternal immune system and releasing HCG to support the pregnancy. To sum up the cells we have seen so far, our fertilized oocyte becomes a zygote. The zygote undergoes several rounds of mitotic division until it reaches a morula, between 16 and 32 cells large. The morula then forms a fluid-filled cavity for which the inner cells are bunched to one side and form the inner cell mass. The formation of this cavity is the blastocyst stage. The inner cell mass are constructed of embryoblasts and the outer cell mass are constructed of trophoblasts. These trophoblasts divide to form the supportive cells, the syncytio trophoblasts and the cytotrophoblasts. The embryoblasts divide into our bilaminar disc, formed of hypoblast, from which we develop the yolk sac, and the epiblast. The epiblast forms an amniotic cavity. Now we have recapped where we have been, let's go and have a look at the learning objectives for this video. After this video and any necessary further study, you should be able to define gastrulation. Explain which tissues and systems of the body arise from the 3 germ layers. Explain the role of the primitive node and streak. Summarise how the left and right body axes are established and the major genes and gene products and pathways involved. List examples of congenital defects that result from abnormal gastrulation and explain their basic pathogenesis. Gastrulation begins in week 3. And the first step of gastrulation starts with the formation of the primitive streak. This image here represents a dorsal view of the embryo at the beginning of gastrulation. We can see that the primitive streak is formed from a primitive pit, a primitive node, and a primitive groove. The primitive pit is the most cranial part of the primitive streak, as we can see, and the primitive groove continues caudally towards the future position of the cloacal membrane. As the bilaminar disc provided us with a dorsal ventral axis, the primitive streak helped to form a cranial caudal axis as a byproduct. Because of these two orientations, we have firmly established a left and a right axis too. So the formation of the primitive streak exacerbates the directions in which organ systems will be developed in a process called fate mapping. Let's go and look at how this takes place. This image represents a dorsal view of the bilaminar disc. The blue giant oval represents the epiblast layer. The right side of the screen will be the head and the left side of the screen will eventually be the caudal end. By the blastocyst stage, the dorsoventral axis is determined and cells are destined to form and cells that are destined to form the anterior visceral endoderm, AVE, migrate towards the head region. At this bilaminar disc stage, cells in the AVE express genes essential for head formation. These include transcription factors, OTX2, LIM1, and HESX1, and secrete factors, cerebrus and lefty, which inhibit nodal activity. Nodal is involved in initiating and maintaining the primitive streak. The absence of cerebrus and lefty at the caudal end of the embryo allows nodal expression to continue, and this signal establishes and maintains the primitive streak. Once the streak is formed, nodal upregulates a number of genes responsible for the dorsal and ventral mesoderm and the head and tail structures. Similarly, HNF3 beta maintains the node and later induces regional specificity in the forebrain and the midbrain areas. Without HNF3 beta, embryos fail to gastrulate properly and lack forebrain and midbrain structures. Please note that there was a typo on this original slide as indicated by the red box. The title of the slide is correct. This is about maintaining the cranial end. Regulation of the mesoderm formation in the middle and caudal regions of the embryo is controlled by brachyury, a chi gene expressed in the node, notochord precursor cells, and notochord. This gene is essential for the migration through the primitive streak. Brachyury encodes a sequence-specific DNA binding protein that functions as a transcription factor. The DNA binding domain is called the T-box, and there are more than 20 genes in the T-box family. Thus, mesoderm formation in these regions depends on the gene product, and the absence results in shortening of the embryonic axis, called caudal dysgenesis. The degree of shortening depends at the time of which the protein becomes deficient. So this slide is new because in the lecture I drew these images on the board. As we have mentioned, the primitive streak forms in the epiblast layer, and this streak allows the migration of epiblast cells to form 3 germ layers. You'll probably remember this from early on in your medicine program. The epiblast cells migrate towards and through the primitive streak. As they make their way through the primitive streak, they displace the hyperblast cells. These migrated epiblast cells are going to form the definitive endoderm. Once enough endoderm has been produced, the epiblast layer undergoes a second round of migration through the primitive streak and forms a middle layer between the definitive endoderm and the epiblast layer, and this forms the mesoderm. Finally, the remaining epiblast layer rests after successive migrations and sits on top of the mesoderm, where it is now referred to as the definitive ectoderm. So that sums up how we establish a cranial-caudal axis and how the 3 germ layers are formed. But what about the left and right axis, which is a process called lateralization? On this slide, we have shifted to a transverse perspective. The left side of the embryo is on the right side of the screen, as though we were addressing it in any transverse view in anatomy. Laterality, or left-right-sidedness, is also established in early development. Normally, many organs exhibit asymmetries, including the heart, the lungs, the gut, the spleen, the liver, the stomach, and others. Positioning these organs and establishing their asymmetries is orchestrated by a cascade of signal molecules and genes. When the primitive streak appears, FGF8 is secreted by cells in the node and the primitive streak, and this growth factor induces the expression of nodal in the lateral plate mesoderm. Nodal expression is then restricted to the left side of the embryo by the accumulation of serotonin on that left side. These high concentrations of serotonin on the left activate, which restricts nodal expression to the left side of the primitive node. Midline genes like sonic hedgehog, and lefty are not only involved in establishing the midline, but are also involved in preventing nodal expression from crossing over to the right side. Ultimately, nodal protein in the left lateral plate mesoderm initiates a signalling cascade that includes lefty to upregulate PITX2. PITX2 is a homeobox containing transcription factor, that is, a master gene responsible for establishing left-sidedness, and its expression is repeated on the left side of the heart, the stomach, and gut primordia, as these organs are assuming their normal asymmetrical body positions. It is prevented from crossing over to the right of the developing embryo, which explains why this organ systems are left-sided. So now we have looked at the mechanism of gastrulation. Let's have a look when things go wrong during gastrulation. Gastrulation is a tightly controlled process and its disruption is very vulnerable to insults. We've seen in some depth that gastrulation forms our body axis, its dorsal ventral axis, its craniocaudal axis, and lastly we have seen how it provides us with asymmetry around the midline in the process called lateralization. Because it is vulnerable to insults, it is possible to see some of these malformations throughout your time in medicine. Look at this CT scan. We can see that there is something wrong with this picture. To orient yourselves, take note of the left and right of this CT, which we are viewing from the feet up at the level of the thorax. This large grey blob in the middle is in fact the heart, but when we look at the orientation, we can see that the heart is pointing in the wrong direction. This is a condition known as dextrocardia, in which the heart has developed abnormally due to a disruption in laterality. Dextrocardia falls under a lateralization defect called situs inversus. Total situs inversus is typically less problematic than dextrocardia because of the switching around of all the viscera, which mean that the great vessels are also flipped from left to right. Thus, despite being a mirror of normal anatomy, it should all work the same. Dextrocardia, however, is only the switching around of the heart. Thus, essentially, we can have a plumbing issue, leading to things like pulmonary hypertension and other associated defects. Now we will look at other gastrulation defects, but please be cautious when viewing the following images in public spaces as they contain fetal specimens with congenital malformations. Gastrulation itself may be disrupted by genetic abnormalities and toxic insults. In caudal dysgenesis or sirenomelia, insufficient mesoderm is formed in the caudal-most region of the embryo. Because this mesoderm contributes to the formation of the lower limbs, the urogenitary system and the lumbosacral vertebrae, abnormalities in these structures ensue. Affected individuals exhibit a variable range of deficits, including hyperplasia and fusion of the lower limbs, vertebral abnormalities, renal agenesis, imperforate anus, and anomalies of the genital organs. In humans, this condition is associated with maternal diabetes. The beginning of the third week of development, when gastrulation is initiated, is a highly sensitive stage for teratogenic insult. At this time, fate maps can be made for various organ systems such as the eyes and brain, and these cell populations may be damaged by teratogens. For example, animal studies have indicated high doses of alcohol at this stage can kill cells in the anterior midline of the germ disc, producing a deficiency in the midline of the craniofacial structures and resulting in holoprosencephaly. In such a child, the forebrain is small and the two lateral ventricles often merge into a single ventricle, and the eyes are close together. Because this stage is reached 2 weeks after fertilization, it is approximately 4 weeks from the last menstrual period. Therefore, the pregnant person may not recognize that they are pregnant, having assumed that menstruation is late and will begin shortly. Consequently, they may not take precautions they would normally consider if they knew they were pregnant. We have already looked at the lateralization of the heart and dextrocardia, so pause the video now and summarise the notes that we have discussed. Sometimes the remnants of the primitive streak persist in the sacrococcygeal region. These clusters of pluripotent cells proliferate and form tumours known as sacrococcygeal teratomas, which commonly contain tissues derived from all three germ layers. This is the most common tumour in newborns, occurring with a frequency of about 1 in 37,000. Teratomas may also arise from primordial germ cells that fail to migrate to the gonadal ridge, and we looked at this during lecture 1. And that brings us to the end of this video. You should now be able to define gastrulation, explain which tissues and systems of the bodies arise from the three germ layers, explain the role of the primitive node and streak, summarise how the left and right body axis are established and the major genes and gene products and pathways involved, and list examples of congenital defects that result from abnormal gastrulation and explain their basic pathogenesis.